Evaluation of a wastewater discharge plume using scanning, split-beam, hydroacoustic techniques, and 3D acoustic tag tracking techniques. This study utilized not only hydroacoustic evaluation and acoustic tag techniques, it also was a very comprehensive study that included a lot of other measurements such as river flow, water discharge, water temperature, fish species that were passing by the study site. It was a very comprehensive study. I'm only going to present the hydroacoustic and acoustic tag results. First, I'd like to acknowledge my co-authors, Sam Johnston from HTI and Dave Thomas from Robertson Bryan Incorporated. The study was conducted for Regional Water Quality Control Board, Central Valley Region, on behalf of Sacramento Regional County Sanitation District. The study objectives were to determine the thermal impact on fish by wastewater effluent from a treatment plant. The methods that were used were a combination of scanning fixed aspect split beam hydroacoustic techniques along with 3D acoustic tag tracking techniques. The study location was in the Sacramento at the Sacramento Regional Water Treatment Plant diffuser pipe, and the study was conducted between February and May 2012. This is just a plan view showing the Sacramento River, and the wastewater diffuser is the line going across the picture. The river flow is from top to bottom. The first 200 feet of the diffuser pipe were blocked. The other 300 feet out here in the river was where the effluent exited the diffuser pipe. And this is a picture showing across the river, and the approximate location of the diffuser pipe is this line right here. You can also see quite a few fishing boats and the fishing pressure during periods within this study period. And this is another slide showing the diffuser pipe location in the evening when there was no fishing occurring. We utilized an HTI digital echo sounder. We used a model 244 multi-frequency echo sounder. We operated it at 200 kilohertz. We utilized two elliptical beam transducers, 4 degrees by 12 degrees, and also utilized two remote dual axis rotators. And this shows where our two transducers were located, and we scanned in 14 different scanning angles from both transducers over an hour period. So we sampled approximately 3 minutes per scan per hour. The minimum on-axis target strength that we were operating at was minus 40 dB. The operating frequency was 200 kilohertz, and we utilized a chirp signal. This technique is described further in Stagg and Johnston, 1996. Then we also utilized acoustic tag techniques. So this is just a picture now with the flow being from right to left of the study site with the diffuser pipe, and the dots correspond to where we installed 14 acoustic tag hydrophones. And for the acoustic tag portion of the study, we utilized a Model 290 acoustic tag receiver and 14 hydrophones. For the predator portion of the study, we utilized a Model 795 LG tag, and for the tagging of Chinook smolts, we utilized the Model 795 LM tags, which are approximately 0.6 grams in weight. All the acoustic tags are fully programmable for pulse width and period, and they operate at 307 kilohertz. So I'm just going to jump into the results. This is, I believe, the only presentation at this conference that utilized single fish tracking techniques, and then from the single fish tracking calculated out the densities. All the other densities for this conference were utilizing echo integration, but this was single fish tracking technique and then calculating out the density from the sample volume. So here's a slide showing the results with the lower densities being the blues and the light greens, and the higher densities being the hotter colors, yellows, orange, and red. So as you can see, there's really three areas of high densities here near the end of the diffuser pipe, and then one other location near the west shore, near the east shore. And this is the underwater view showing those areas of higher concentration here along the east shore and near the end of the diffuser pipe. So now let's go into acoustic tag position results. So this is a slide that is an animation, and it's going to show the tracks from the first two release groups of Chinook smolts. In each of the group, there were 75 fish that were tagged and released, and they were tagged approximately 10 miles upstream. What you're going to see are the tracks going through the study area with color corresponding to the elevation of the fish, with the reds and the oranges being deep in the water and the yellows and light blues being mid-water column and the blue being near the surface. And they're going to pass by very quickly. And in fact, this is approximately 2 weeks worth of data that will be shown in approximately 20 seconds. So you can see, for the most part, the path of these fish were straight downstream. So that was the first release group, a couple stragglers. And now here's the second group of 75 Chinook smolts migrating downstream. So you can see this is now just showing all of the first group of data. And as you can see, they're very straight path going by the diffuser site. The concern was that there would be a lot of predation right at this site. And so, at least from our results of the tagged Chinook smolts, we saw no predation occurring at this site. We also tagged 99 predators, and these predators range from striped bass to pike minnow to American shad to smallmouth bass and white catfish. Of these 99 predators, there were only 52 that even stayed in the study area more than 1 hour, and of those 52 predators, there were only 7 that stayed within the area for any length of time on the order of a few days. So this is just a time series showing the tag codes and the study period and showing the periods when the fish were detected in the array. So you can see, for the most part, there's only approximately 7 fish that stayed in the area for any length of time. Since we know the tag position and we know the amount of time that it stayed in the study area, then we can calculate out a position density. And that's described by Stegman and Johnston 2010 publication. So as you can see, again, the reds and oranges being high densities, the light greens and blues being low densities. So you can see where the high density of these fish tagged predators were. 2 of them near the east shore and the other 5 being near the end of the diffuser pipe. So if you looked at this in plan view, again, you can see here 2 of the fish that spent quite a bit of time near the east shore and the other 5 near the end of the diffuser pipe. Very similar to the hydroacoustic results. One thing that we noticed near the end of the study, we had some equipment problem where the fishermen actually drug their anchors and damaged both of our hydroacoustic rotators. And so we had to pull them out, and it took a couple weeks, and the study was supposed to be completed by this time, but we got it installed, and so we were supposed to monitor another couple weeks, so we did. And it happened right during a period of the American shad migration, upstream migration. And so during this period, there was quite a few fishermen in the area. And so there was a little concern about the American shad reaction to 200 kilohertz hydroacoustics, but there's nothing in the literature regarding American shad. There is studies that have shown European shad avoid 200 kilohertz sound. Jim Gregory published that probably about 5, 6, 7 years ago, but nothing with American shad. And so what we found, which was very surprising to me, was that we had very high densities of fish, but it was really congregated at a range of greater than 20 meters and in a range band of approximately 6 or 7 meters. And so I believe what this was that the shad just didn't want to get any closer than 20 meters to our hydroacoustic 200 kilohertz sound wave. And so regardless of what aiming angle we had, they would stay that distance away from the transducer as they migrated upstream. Also, at the end of when we pulled out the equipment, we found that the fishing was poor after this period of time, and so the fishermen went away soon after we turned off our hydroacoustic system. So, in conclusion, there were 298 Chinook salmon smolts that were acoustically tagged and tracked passing the study area of the wastewater discharge pipe. There were 99 predator fish captured and acoustically tagged and tracked swimming in the area of the wastewater discharge pipe. The split-beam hydroacoustic showed high areas of density predators, primarily in 3 distinct areas downstream of the wastewater discharge pipe. The split-beam hydroacoustics appeared to show the behavioral avoidance of American shad to the 200 kilohertz acoustic beam during the last 2 weeks of the study, with the highest density of fish approaching no closer than approximately 20 meters from the